Hello class, this is Mr. Roth here. Today's lesson is going to be on enzymes. Enzymes, although seemingly very small and unimportant, really make up the entire function and of our entire bodies. Without enzymes, life would come to a grinding halt. So what are these molecules? What do they do? I'm going to give you some examples. But before we start, I do want to remind you of the central dogma of biology. Central dogma is just the big idea that it's our DNA ultimately that's responsible for all of the physical and tangible stuff that goes on with our bodies. You'll notice that here in this image we have a picture of our double helix DNA. And of course the process where we move forward to get RNA is called transcription. Once we have that RNA strand, translation will then make the tangible and physical protein. A lot of people think that our bodies are made of DNA. It's not true, actually. DNA is stored inside the cells, but most of our body, in terms of the substance, is water and proteins. It also turns out that these proteins are, in fact, what are in charge of all of the happenings inside of our cells and inside of our bodies. They're in charge of the machinery that makes life go on. So proteins, and particularly enzymes, are arguably the most important macromolecules in all of biology. Our physics teacher likes to joke that whenever there is something that we don't know, we just say, well, the enzymes do it. And to some extent, saying that is true, but what he's mocking is that we don't really know how they do it. If you look down here, it shows that genes code for some sort of protein, always. Every gene does. Some examples of different proteins would be collagen. Actin and myosin are the proteins that make up our muscles, and then things like repressors that control the cell cycle, along with enzymes that catalyze reactions. When we look at what an enzyme does, what I'd like you to first pay attention to here are the two different colored graphs. You'll notice that the graph with the red line is basically showing a reaction without the enzyme. We look at the blue line, and this is the reaction with the enzyme. The big difference, you'll notice that both of them actually do happen, right? They both, the same reaction occurs in both. But it turns out that without the enzyme present, the amount of energy needed to get over the hill and then finally proceed with the reaction is significantly higher. And this is really what an enzyme does, is it catalyzes. There's a new word here, catalyze. And catalyze just simply means helps the reaction proceed. The way that it does this is it lowers the activation energy, bringing it down to a more feasible hill to get over for the reaction to proceed. Okay, so first, before we talk about some real-life examples of enzymes, I'd like to give you enzyme terminology so that we can begin our discussion. Every enzyme has something called an active site, and it's really just a very specifically shaped pocket that is designed to bind with a particular molecule. In this case, the molecule is shaped like this, and so likewise the active site is shaped accordingly to bind it. Once that substrate, and let me define substrate. Substrate is the thing that is being reacted. And I will give you some examples of those later. Once that substrate binds and is connected, we call this an enzyme-substrate complex. One other thing I wanted to point out too is that enzymes, their names almost always end in ASE. So you think of the enzyme catalase, lipase, lactase, which is a very important enzyme to help us digest milk. Lactose intolerant people don't produce enough of this. And then lastly, polymerase. We've already talked about this enzyme, which helps to polymerize. In other words, it connects together monomers to form polymers. Okay, so here's our first example. It turns out that our cells produce hydrogen peroxide as one of the byproducts of many of the reactions that our cells do. However, hydrogen peroxide is toxic in large quantities, and in fact, that's why we use it as an antiseptic. Have you ever seen the big brown bottles of hydrogen peroxide that you can use to sanitize your wounds? Well, the reason that it sanitizes is because it is toxic. So what the cells have done is they've come up with a way of getting rid of it, and so they synthesize an enzyme called catalase. Catalase will cause hydrogen peroxide to decompose into two products. And the products that it decomposes this molecule into are water and oxygen, which happen to be actually useful to the cell. So we're going from something harmful to the cell to something useful. So this is a very adaptive enzyme to have present. So it turns out almost every cell that exists on planet Earth produces catalase. It's necessary for the survival of the cell. So if we were to look at this, if we could zoom in on the reaction and how it happens, we would see something like this. This is a cartoon illustration, but it's pretty accurate. You'll notice that the purple is our enzyme, and present here would be the active site, which is shaped perfectly to bind the substrate. The substrate in this case corresponds to hydrogen peroxide, and then the two products that come out are going to be, in this image they're showing glucose and fructose, but ignore that for a second. The model still works for our reaction. In our case, this product would be water and this product would be oxygen. So really what the enzyme does is it splits apart the hydrogen peroxide into two products, hydrogen and water. Okay, so here is a picture of it. If we were to actually conduct the experiment, we would need first a source of catalase, which really, you can use yeast, you could use liver tissue. Those cells produce catalase, and so really you're just putting some living tissue, mixing it with hydrogen peroxide, and it's going to bubble. In fact, if you have an open wound and you pour hydrogen peroxide, the cells that are in that open wound, even in the blood cells, contain catalase, and so you'll see it fizz, you'll see it bubble. What I'd like to ask you is, what do you think the bubbles are made of? If we could test the gases that are in these bubbles, what would be inside? Well, if you remember, our prior reaction here showed that oxygen was the gas that's being produced. So if you guessed oxygen, you're right. The bubbles here are oxygen bubbles. I'd like to give you another example. We had mentioned lactose intolerance. Now at first glance this reaction looks very complex, but try to ignore all the fancy letters and just look at the shapes and the bonding. You'll notice that we have a disaccharide, di meaning two sugars connected together. That's what lactose is, and it's the type of sugar that milk is made of. We find this sugar present in milk, and for our body to actually process these sugars and go through cell respiration, we first need to break it down into its monomers. And so our body has an enzyme called lactase that is specifically designed to do such a thing. We look over here, and on the products side of the reaction, we find that what it comes out are two different monosaccharides, both of which can be used by the cell to produce energy. So if we look at the cartoon version, we see again our active site binds the sugar, reaction happens, and the product is that the disaccharide is split into two monosaccharides. With a lactose intolerant person, there could be two causes that are causing them to fail to digest the lactose. One of them could be that if they have a mutated gene that codes for that lactase enzyme, you're going to end up with a misshapen or denatured protein, or lactase enzyme. And so the result of this can be seen down here. If you look at the active side of this denatured enzyme, you notice that it does not in fact correspond or match up with the lactose sugar or substrate. So if it doesn't match up, it's not going to bind and it's not going to react. And what this can result in is, in a person with lactose intolerance, they can't digest lactose. and it can have a number of unpleasant side effects like bloating, diarrhea, etc. Interestingly enough, though, there is a product out there called Lactaid, which is a tablet with a key ingredient to help people digest their lactose. And the key ingredient, as you might guess, is a non-denatured or a healthy lactase enzyme. So it's a lactase supplement, and it helps to digest the lactose. In certain cases, if the conditions are not right, the shape of the enzyme can actually change. And this has nothing to do with mutation. This just has to do with the wrong temperature or the wrong pH. So if we look at the graph down here, we notice that there is in fact an optimal temperature, and it appears to be in the range of 35 to 40 degrees Celsius, which corresponds with our body temperature. We look at this and we see that if we get on the too cold side, we notice that the activity of the enzyme decreases. And that simply has to do with a lack of molecular motion. Cold temperatures basically slow the molecules down. But if we go on the other extreme and we get toward the high temperatures, what we're showing here is that the enzyme will change shape. And again, just to remind you, if we change the shape, we can no longer have that substrate bind and the reaction will not proceed. And so, to get the highest level of activity, there's an optimal temperature for every enzyme. We could look at the graph for pH and it would actually resemble this graph almost identically, where there is a peak and then lows on either extreme. Okay, folks, that is it. I hope that enzymes are making sense. We will be doing some experiments on them in class. Looking forward to it. Adios.